the genetics of cancer, signal transduction pathways in the development of cancer. Signal transduction is one of the most important process by which a cancer develops. Now, signal transduction is a process of conversion of external signals such as growth factors, cytokines, hormones to a specific internal cellular response such as gene expression, cell division, or even cell suicide. This process begins at the cell membrane where an external stimulus initiates a cascade of enzymatic reaction inside the cell that typically includes phosphorylation of proteins as mediators of downstream processes. Therefore, identification of pathways, that is signal transduction pathways, is very important for research on drug discovery and to take some therapeutic measure. So this is also true in case of cancer. So in this module, we'll discuss about these signal transduction pathways in the development of cancer. In case of growth factor receptor pathway, this is one of the most important thing, that is receptor tyrosine kinase, which is present in the cell membrane. Now, RTK or receptor tyrosine kinase, it has two ends, N-terminal end, which is located outside the cell membrane. We are showing it over in this figure, and C-terminal end, which is located inside the cell membrane. When ligand binding occurs, some growth factors are there, as for example EGF or epidermal growth factors, PDGF, platelet derived growth factor. Now, when the ligand binding occur in the N-terminal end, this is the N-terminal end, when ligand binding occurs, dimer formation takes place, that is receptor dimerase. After this dimer formation, there is a autophosphorylation occurs. So here we are also showing, that is autophosphorylation occur at C-terminal end. So after autophosphorylation, this signal will pass. Secondly, if the receptor tyrosine kinase is mutated, then oncogenicity occurs. Here in this figure we are showing, that is here this receptor tyrosine kinase, where N-terminal and C-terminal ends are truncated or deleted, as a result of which there is a constitutive dimer formation occurs and autophosphorylation sets in. So it is not under any control and constitutive autophosphorylation occurs and signal passes. So this occurs in case of mutant receptor. Now let us see how these pathways, how this signal passes to the transcription factors and how gene expression occur. One of the most important pathway is kinase pathway. In the kinase pathway, in this figure we are showing, that is there is a receptor tyrosine kinase or RTK. When a growth factor interacts with this RTK, phosphorylation occurs, and this signal passes to one important protein that is called RAS protein, which is activated when it bind with guanosine triphosphate. So GTP activated RAS protein transfer this signal from receptor to some kinase, cytosolic kinase proteins. One is your RAF, MEK, MEK, and MEK kinase. Now cytosolic signals will pass to the nucleus and it activate the two genes. One is Jun, another is FOS, Jun and FOS. Now the Jun and FOS, it will synthesize a dimeric protein that is API, apo-inducer, API, that is apo-inducer. Now this is a dimeric protein. One monomer is synthesized by Jun, another monomer is synthesized by FOS. This dimeric protein, that is apo-inducer, API, is a transcription factor. It recognizes a sequence that is TGA, CTCA. This is the sequence it recognizes on the target gene and activate the target gene. So API acts as a transcription factors. It recognizes a sequence, this TGA, CTCA, and activate the target gene. So if there is any mutation and in Jun and FOS, as a result of which there is a abnormal growth or uncontrolled cellular proliferation occurs and cancer develops. So this is the one of the kinase pathway. Here we have shown that is how signal is transferred from receptor to the nucleus and how your target genes are activated. Another pathway is also there. This is also a very important pathway and very simple pathway, better to say. That is called JAK-STAT pathway. So in this figure, let you see one cytokine receptor is there where a cytokine binds. When a cytokine binds, this cytokine receptor, a dimer formation occur. After dimerizations, another kinase is there that is called JAK kinase or Janus kinase. This JAK kinase or Janus kinase is a type of tyrosine kinase. This has two domains. This is called kinase domain. These two domains we are showing over here. Now this activated or kinase domain, it binds with dimer, cytokine dimer, there is cytokine receptor dimer. After binding, it has a substrate, it is called STAT. This is the one transcription factor, the STAT. So STAT will also bind and with your Janus kinase and cytokine receptor, and they form one multi-component body. And here the STAT become phosphorylated. STAT is phosphorylated. So here STAT is phosphorylated. So phosphorylated STAT now translocate to nucleus. It will translocate to nucleus and it will activate a transcription factor. So here we can see that is the JAK-STAT pathway, which has got only three component, JAK, STAT, and one cytokine receptors. And signal is passes and gene is activated through a transcription factor. So in this module we have discussed about the signal transduction and pathways responsible for the transfer of signals, kinase pathway and JAK-STAT pathway. We have also provided you key concepts of the module, list of books for your further reading, some quizzes are there, and study material for download. So you are also feel free to ask any questions if you have. Thank you for participation in our online course.